that it will record. Aha, you are recording, it says. Now that's good. And now I'm heading over to share screen, start broadcast. And if everything goes well, hopefully, can you see my screen? Can you see my screen? Yes. Yes, the spiders are coming out. That's great news, isn't it? I missed them. A friend of mine recently complained that ladybirds are coming out as well. Well, poor ladybirds. I think it will get colder probably next week, but for the time being, let's just simply enjoy things. So good morning, everybody. Good morning, good morning, good morning. Lovely to see you all. I think it's a lovely community, although we don't see each other in person. But I really love these sessions with you guys. No, I think the brief has been given not to include the material from a week in which then on the Friday the program level multiple choice quiz will be. So I try not to include the material that I do, say, today and on Friday in the quiz on Friday. Unless you want me to. Do you want me to? The dogs are fine, although one of them was a bit sick this morning, but then she's going into season, and therefore it's a little bit of an issue. So she's a bit fractious at the moment. Yeah, I get the message. Hey, don't worry, I'm not that cruel. I'm not. It's not, contrary to popular belief, I don't get any pleasure out of torturing students. I can't answer this question because I have nothing to do with WhatsApp. I use it, but not for teaching. There is some private space, but there is a Facebook group chat. Okay, guys, let's get started because we've got a lot to do today. I don't torture students. Well, some students might disagree with you on that, Reynolds. Right, let's just get started. Now, last week, was it last week? Yes, last week, we discussed how we can actually regulate enzyme activities. And we discussed numerous ways how enzymes can be actually be controlled. We discussed the concept of effectors. We discussed, for example, how inhibitors or activators can do something with enzymes. We said, for example, substrate can be an activator. when there is this concept of cooperativity, when the subunits of an enzyme talk to each other, and some people referenced hemoglobin and said that they've encountered that before. So we discussed that. We talked about modifications, and very often these are post-translational modifications. So post-translational modifications of the enzymes. For example, phosphorylation of an enzyme. So the enzyme becomes phosphorylated, which brings the enzyme from an inactive state into an active state. Or the other way around, from an active state into an inactive state. We said that the redox environment can modify enzymes. One thing that I forgot to mention, or I didn't have time to mention, is that occasionally we find something like partial proteolysis. Partial proteolysis of an enzyme. And we find that very often when we are dealing, for example, with for example, digestive enzymes. So digestive enzymes, enzymes that break down proteins or DNA or RNA or anything else. And it would be absolutely devastating for a cell if, for example, a protein degrading enzyme was active inside the cell, because it would destroy everything. So what happens very often is that these enzymes are produced it would be absolutely problematic. It would be the end of the cell. So these enzymes are produced as inactive precursors. So the enzyme might look like something like that, with this bit sticking out here, and again that is part of the protein. So that's inactive. And then when we need the enzyme, we just simply chop off this bit here. So we get the active enzyme plus this precursor part here that I've drawn here. And now the enzyme is active. And because we chop off or we hydrolyze a peptide bond, this is called partial proteolysis, where we just simply chop off a bit that is part of the precursor protein, and now we have the active protein. And if you're missing Vmax, don't you worry. And this here is just a pair of scissors that I indicated here. Maybe. Yep, I think that's a lovely definition. So we hydrolyze this peptide bond between the enzyme and this precursor, and this is a breakdown of a protein bond, basically. Partial proteolysis. We don't totally chop up the enzyme, we just break this particular bond. Okay, so these are all ways how we can actually regulate enzymes. But if you think about it, enzymes are just sort of the smallest units of what's happening in the cell. Let's have a look at a scheme, what is actually happening in the cell. And people are, I know that you have been waiting for that. We are now moving towards the metabolism part of this module. So let's have a quick look at what's going on in the cell. Whoa, look at that. This is just a little bit of an overview of what is happening in the cell. I personally think this is an sort of oversimplification of the processes that are happening. But you know, we will go into more details. And by the end of this module, you will not have any problems identifying these components. No, I'm not having a laugh. So you will immediately see that, for example, this bit here, that's the citric acid cycle. Here we have a breakdown in terms of beta oxidation of fatty acids. You, without any problems, will identify the components of this pathway here. So that's glycolysis. So there is quite a bit of stuff going on. So yeah, I think actually this company that produced this chart, I think they are still producing them, and you can get them as A0 charts. I got some, and I put them up in the downstairs loo. So it's really great stuff. Oh, it's a funny looking tube map. This is called metabolic or biochemical pathways. I think I can, you can Google that and you can have a look at it. So yeah, it's a funny tube map. Actually, you say that. It does look like a tube map, doesn't it? And we can easily read tube maps. That's the map of the London Underground. And therefore, you know, something like that is a little bit more information there than in this tube map. But, you know, basically, it's pretty much the same thing. So how is this actually... how do these reactions actually fit together? Because that is what we really want to understand in metabolism. So we usually look at things as sort of sequential reactions. So we start with a reaction with a compound A. So in this case here, I've chosen arbitrarily, let's say, glucose. And then we have a sequence of reactions. And eventually at the end, we come out with a modified compound. In this case, this would be pyruvate. And glucose is basically converted into pyruvate. And we are going to discuss that, the principle of glycolysis, in more detail. What I just want to show here is that we have just simple sequential reactions, that one reaction follows the other. And we can even figure out what the order of these reactions is. And that is actually one of the tasks. This is one of the tasks in the BI301 practical, where we need to figure out what is actually the correct order of certain reactions before we reach, let's say, this endpoint E here. And the way we can do that is we can almost think about it like the tube again. Let me go back. So let's say we want to figure out how many stations are before this station if we start here. And the way you can do that is you just simply get people on the tube here, and then they are traveling along here. And the further away a certain station is, the longer it will take people to get to this point. And that's pretty much the same thing. Yes, that's the one. Thank you. We can do exactly the same thing. What we do is we just, for example, we add the different components at each step, and measure how long it basically takes to reach this point E. So we take basically all the enzymes together, but no substrates. We add A here, this component, and after a short period of time, we measure how much of this component has reached E. And it will be, not much of A will have reached E in, let's say, 5 seconds or so. But if we start with the component D, almost all of D that we add will have reached E in 5 seconds. So what we can do is we can sort of create an order in which these components are. And you probably noticed I deliberately made a mistake so it doesn't go A, B, C, D, E. In this case, I deliberately put C before B because that just simply indicates that, these components have a defined order, and sometimes the order is not necessarily what we would expect. Now, there is an exercise in the BI301 practical that you have to do. And there is also a little tutorial where you have to find out the correct order of such a sequential reaction. But of course, these pathways can also be, in a way, branched. So we could go here another component, EF, and then we might have a component G, and so on and so forth. So it doesn't have to be just a straight line. There could be branches, like, again, we have with the tube, where we have certain junction points. So, for example, King's Cross, or with platform 9¾ and other Victoria, they are junctions, and we have the same in biochemical pathways. So we can look at pathways this way. Now, we already, in the BI308 module, we discussed the concept of a steady state. And that is something that we will find very, very often. Basically, in all biochemical pathways, we find this steady state concept in these biochemical pathways. And steady state, this is a little bit like with a tap or something like that. So you've got a tap here, and if the influx of water is the same as the outflux, what will happen to the water level here? And I've put that down here. The water level doesn't change because what goes in comes out again. And that is the principle of the steady state concept, which we have for many, many biochemical pathways. We have an influx, and if this influx is the same as the outflux, or flow out, flow in and flow out, then the concentrations of A they will stay constant. Like the water level. Right? Good point, Reynolds. Is it an equilibrium? The answer is well, in an equilibrium, we would have A, B, right? That would be an equilibrium where eventually the concentrations don't change when we reach the equilibrium. However, here we don't really have an equilibrium because it goes in, so that the compound will move that way. So it's not an equilibrium, but it is an equilibrium. It's sort of a pseudo-equilibrium, right? But the concentrations of A and B will stay constant. So it is an equilibrium, but it's not really an equilibrium. But it's an equilibrium. I know I sound like Boris Johnson, go to work, but don't go to work. But it is complicated. Yeah. But the concept of equilibrium is really, it sort of applies, but we need to be careful. It's an equilibrium is when we have a closed system, but here we have an open system. So as I said, an equilibrium is closed, but here we have an open system, and therefore technically it's not an equilibrium, but it behaves like an equilibrium. And the open system means material comes in and material goes out. Okay? And that's exactly, that sounds absolutely right, Edith. So in an equilibrium, we have the compounds staying the same in a steady state. Once we have reached the steady state, again, our components A and B, so A is this one here, A and B, the concentrations will stay pretty constant. Absolutely right. Yes, that's a good definition, Jerry. So our concentrations of A and B stay constant, right? If we have this steady state. Now let's have a look at some interesting cases. Please bear this graph in mind that we have here. This one here, yeah? Bear this in mind. It's not terribly complicated, bear it in mind. Right, now, here we have now a system which is pretty much the same as we had before, but we have our component B, this component B, acts now as an inhibitor for, let's say, an enzyme that produces A. Right? So we have a steady state, so we have influx here, and we have outflux, so in and out. But in between, we have B, this component B, which inhibits the production of A. What do you think will happen? Will we get, again, some kind of steady state levels? Constant concentrations of A and B. What do you think? Well, B is getting out because we've got outflux as well. So let's think about it. At the beginning, we have no B. At the beginning, we have no B. So A is produced, A produces B. B now stops the production of A. That's this one here, that's indicated with that. B stops the production of A. So A does no longer produce B. But then B is taken out of the whole thing. So if B is taken out, it can no longer inhibit A, so A starts producing again. The yellow area. Oh, forget the yellow. That's just the background from where I took the screenshot. Will we reach a steady state? Himat, will we reach a steady state in this case? I hope so. Let's have a look. What happens? Hey, look at that. At the beginning of the reaction, we see a little bit of what is called an oscillation, a bit of fluctuation, but eventually what we get is, again, constant concentrations of A and B. So we reach a steady state. A, great. Yep. And that is the typical characteristic, or that's the definition of a negative feedback loop. And typical for negative feedback loops, result in, they make a system, they make something more stable, actually. More stable. They keep the system sort of at the right level, even if we change the concentration of A, then we increase B and we get everything back to normal. And a very important thing is we will always get the same ratio of B over A, the same concentrations. And it does not matter what our starting concentrations are. So let me write that down. Does not matter what the starting concentrations are. We will always get the same value for the concentration of B over A in this steady state. Okay, does that make sense? So that's a negative feedback loop, and we will see that time and time again when we look at biochemical pathways. Great stuff. Now, let's have a look at another scenario. What could we do? Oh, yes. If there is a negative feedback loop, we can have a positive feedback loop as well. So here we've got our simple scenario with producing A in and out from B. So this is what we had before. But this time B is an activator. It activates. It activates or has a positive effect on the concentration of A. Will we get a stable, steady state? What do you think? Will we get a positive, steady state? No? So we start with A, we produce B, and as soon as we produce B, it will stimulate the system to produce more A. So more A produces more B, which produces more A. So it's almost like an avalanche. It will keep increasing. What will we get? So people say no stable, steady state. And you are absolutely right to a certain extent. Because what we will get when we do a simulation, and there are simulation programs out there, what we will get is something like that. Whoa, look at that. What have we got here? Here we plot the concentration of A on the y-axis and the concentration of B on the x-axis. And what we find is, I need to change the color. What we find that the system actually has not one stable steady state. It actually has two stable steady states. It can either reach this lower state with very low concentrations of A and B, or alternatively, it can reach a very high steady state for both A and B this way here. It depends on the system. Sometimes we have a negative feedback and that stabilizes it. In a positive feedback loop, we can get two different steady states. And these steady states, it depends which steady state we reach, because, let me write that down, which steady state is reached depends actually on the starting concentrations, of A and B. Let's have a look at that again. Here we said in a negative feedback loop, it does not matter what the starting concentrations A and B are, we will always have one steady state. In the positive feedback loop, it depends. If we've got enough A to produce a high level of B, then we will have this avalanche system. But if we don't get enough B and B is immediately destroyed, then it will stay at a very low level. You will get these slides, and I will put up the slides on the class material in Teams, so there is no problem. You don't have to take pictures or anything like that, and I will upload it later. So if we have a positive feedback loop, we get two steady state levels. One of the really interesting cases of a positive feedback loop is prions. With prion diseases like BSE or Jacob-Creutzfeldt syndrome, we have exactly something like that. If you are very low in A and B, nothing happens. But if your concentration of A goes up, then B goes up and you start this vicious circle and you develop a prion disease. Positive, absolutely. In a negative feedback loop, the starting concentrations just simply don't matter. In a positive feedback loop, it is important, the starting concentrations of A and B are absolutely crucial, right? So just by changing from an inhibitor to a positive effector has a dramatic influence on, for example, a pathway. Okay, now let's have a look. Hey, let's look at that. What have we got here? We have one pathway, and the compound A inhibits the production of B. And in another pathway, the compound B inhibits the production of A. Will we get a stable, steady state like we had with a negative feedback loop? What do you think? Or will we get something weird like we had with a positive feedback loop? Nothing will happen. Oh no, something will happen. Makes it stable. Shall I show you what happens from the simulation? Let's go back to this one. Look and try to memorize this picture with A and B, right? Look at that. In this system, again, we have two stable, steady states. But this time they are absolutely opposite. We have high A, but a low B. Or, in this case, we have low A and a high B. In the previous case, we have low A, low B, or high A, high B. In this case, where we have this mutual inhibition, we have, as I said, high A and a low B, and it makes sense. If we have lots of A going through that, it will shut down this pathway. So high A, low B. And if we've got lots of B going through that pathway, it will shut down the A pathway. So we've got two stable steady states, and again, depends on the starting concentration. Okay? So that is mutual inhibition. And we see that all the time. One pathway shuts down the other one and the other way around. Is it gone? Is my screen still gone? Screen is still okay. No, it's there. Yeah, I don't get a notification that it's gone. Okay. This is just a principle that can happen in any pathway, right? I just want to show you some mechanisms that we might encounter. Okay, now let's do something. Let's ramp it up a little bit. Here we have not just two pathways, we have three pathways with negative feedback loops, as we had before. What will happen? One stable steady state or two stable steady states? What happens? One stable steady state or two? So we have got one pathway, A inhibits the production of B inhibits the production of C inhibits the production of A. 1, 2, 3. Are you ready for a shock? I'm going to shock you now. Something that you don't expect. And if you tell me that you did expect it, then you are lying. Because what will happen is this one. We will get an oscillation. And yes, there are stable steady states, but not in time. So the level of A, B, and C will oscillate. Very often, you see this pattern when you, for example, have something that has a time component in it, i.e., let's say your sleep cycle. Most students, the sleep cycle is messed up, but this is usually caused by a three-component system. Menstrual cycle shows a typical oscillation pattern. Cell cycle in general is a typical pattern that is generated by a three system. Absolutely, yes. So we have these three components and they lead to an oscillation pattern. How cool is that? Here's another system that actually puzzled scientists for many, many years. And that is something that I really need you to understand. This was discovered quite a long time ago, where we find that we have a substance or compound A, and this compound A, it can be a protein, it can be just simply a chemical compound, is then phosphorylated. Like we discussed last week with the modifications, it can be an enzyme, and the modification is carried out by a kinase. And so kinase is an enzyme that attaches a phosphate residue to either a chemical compound or a protein, and it uses ATP. So we get phosphorylated compound. And at the same time, people noticed that for each kinase, there is also a phosphatase that dephosphorylates the compound B. So it removes the phosphate group and reverts the whole thing back into A. And if you think about it, what this is it looks like we're just wasting ATP, because here from A to B, we use ATP, and then we bring the system back. And what we've done is we've used precious ATP. And it happens all the time. It goes round and round and round and round. And the effect is we are wasting ATP all the time. And that is why the scientists originally called this a futile cycle. It's a futile cycle because it doesn't do anything. And then scientists realized, actually, hey, wait a moment, there is far more to it. There is more to it than the eye actually needs. Because what happens is, if both kinase and phosphatase, the activity is very different. If the kinase activity is much larger than the phosphatase activity, all of our A will be in the B form and the phosphorylated B form. So kinase activity larger than the phosphate. So for example, Vmax of the kinase is bigger than the phosphatase. Then everything is in B. Because this part here is really strong and the lower part can't cope with it. So we are in B. What happens if the phosphate activity is stronger? What happens if phosphate is stronger than the kinase? Where do we end up? If the phosphate is larger, where do we end up? In A. And actually, you're absolutely right. We end up in A. And actually, what it really depends on is what is the ratio between kinase and phosphatase. If it is exactly the same, we don't know. But if we just are a little bit stronger with the kinase, we are all in B. If we are just a little bit stronger with the phosphatase, we are all in A. And this just a little bit, this is denoted here in this diagram. So here, the yellow one is the B. We've got B here. If the kinase is stronger than the phosphate, if the phosphatase is stronger than the kinase, our concentration of B is low. But as soon as we get to this point where they are both the same, all of a sudden we jump up. This is called zero-order ultrasensitivity, and it is a switch. The system can very easily switch between kinase and phosphatase. All I need to do is increase the Vmax of this one just slightly, and I will end up in B. And if I just increase the Vmax of the phosphatase slightly, I will end up in A. And that is a very, very frequent switch-like pattern. I can switch on a pathway just by modulating kinase and phosphatase activity. And that is one of the things that I want you to really understand from today's lesson. Here's another thing. We can actually inhibit the kinase activity. We can add an inhibitor of the kinase activity. And what will happen if we have the inhibitor present, and I indicate that here with a 1, inhibitor is present, so kinase activity is inhibited. What happened to B? We don't get any B. So inhibitor present, no B. If the inhibitor is absent, we will get B. And I've denoted that with 0 means not there or absent, and 1 means present. So what we have here is what's called a NOT gate in electronic terms. Inhibitor present, no B. Inhibitor absent, yes B. a NOT gate. That's electronic speak. Actually, what we can do is we can combine two of these gates, and we can create what is called an AND gate. Computer science, yes. Electric engineering, we have an AND gate, just with enzymes. Hey, we can create an OR gate, where we either have an inhibitor here, or we have an inhibitor here, and we check what we get out here. No, you don't need to be able to identify these. There is a reason why I show you, and I'll come back to that in the next 2 minutes. But what I really want you to know is the function of this switch that I showed you with the kinase and phosphatase. Where's the squiggly line? So we have AND, NOT, OR gates, and Bernardo said computer science or electronic engineering comes to mind. I hope it's not PTSD, but if you look at that, this is a typical electronic network that we have here, right? With lots of different components in it. Lots of different components. Can we easily manipulate these components? Do we understand, say, an electronic network like this? Do we understand that? Well, maybe not me, but do people understand these electronic networks? Tell you what, my dad, he is an electronic engineer. He looks at this thing and says, yep, absolutely, full get it. And what he can say is, for example, if this thing doesn't work, and I have no idea what it is, it doesn't seem to be terribly complicated, but if it doesn't work, my dad goes along and says, oh, okay, let's measure something here, let's measure something here, let's measure the voltage here, and now I know that this component here doesn't work. My dad does that, or used to do that for a living. He could repair basically anything electronic. Now look at that. This is an electronic network. And let's look at this one. This is a biological network. It is exactly the same because it is made up of exactly the same components. OR gate, NOT. We have resistors in it. We have little batteries in our biological network. So biological network is pretty much the same as an electronic network. So if we could model biological networks with this incredible complex behavior, if we could model these on what we easily understand, and I mean, we can build computers, we can build smartphones, we can build even artificial intelligence and all that stuff. But if we just sort of transfer these components that we are used to use every day from these electronic circuits into biological networks, into something like that. All we need to do for diseases is we measure a component here, we measure a component here, we measure a component here, and we know then that this particular enzyme that catalyzes this reaction here is messed up. So we can replace this enzyme. So hopefully what I learned today is that in biochemical pathways, we have different components in them, but these components are exactly analogous to components in electronic circuits. Now, on Friday, we will discuss what drives these biochemical networks. And what I want you to do is I want you to revise your knowledge, revise a little bit about mass action ratio, and the principle of Chatelier for Friday, because we are going to use that and expand on this analogy of the electronic networks. So I hope you found that interesting and thought-provoking. Biochemical networks are just like electronic circuits. Electronic circuits we understand, and now we can actually use these components to understand also complex biochemical pathways. And yeah, if you did that, BI301, Chatelier's principle, mass action ratio, you probably also have done delta G. Have you done delta G? Because a few people have told me that they haven't. So yes. So have a look at these things. We will very briefly go over them on Friday. And don't worry too much about the maths, because I will try to do it in a gentle form. Thank you very much for watching, and I hope you found this interesting and sort of got really excited about what we are going to do with the metabolism part. Thank you. Have a lovely day and enjoy yourself. I will put up the slides and the stuff on these class materials in Teams, but also probably on Moodle at one point. Thank you and enjoy the rest of your day. Take care. Bye-bye.
Thank you, sir.
My pleasure.